Hi everybody, and welcome to the second Oxford Materials Wednesday webinars. So I will now introduce our speaker for today's session, Dr. Alex Sheader, who is going to talk to us a bit more about electron microscopy and how we use that to image very, very tiny things in material science like cells and viruses. So Alex, I will hand over to you. Thank you.
Thanks, Helena. Right, well, thank you so much for coming and giving up part of your Wednesday. It's really nice to see so many people online. So my name is Alex, and I'm going to talk a little bit about how we use electron microscopy, and particularly very high resolution electron microscopy, to look at biological materials. So everyone, I think, probably is familiar with a microscope that looks like this. So it's a kind of benchtop optical microscope. Very familiar, you've probably used one in biology, maybe in chemistry. Everyone is hopefully familiar with this sort of microscope, and you've probably even used one yourself at some point. So a brief refresher on how microscopes work just in general. Normally you've got an illumination source, and in an optical microscope it's photons that you fire up through your sample. There's a series of lenses made of glass that are in there, and they help focus the light down onto the sample and collect what's transmitted through. And with that, you can make some really nice images. For example, these ones. So this is where the Q&A comes in, actually. So has anyone got any idea what A and B might be? What types of cells are they? Cool. So we've got a plant and animal. Someone else says think it's a plant cell. Oh, excellent. We've got a couple of answers as well. A is an onion, which is exactly right. It's onion skin. It's one of the favourite ones for looking at in biology because they always prepare really nicely. Any ideas for B? Someone who posted in the Q&A is correct that it's definitely an animal cell. Any ideas from where? Okay, so actually kind of similar to the onion skin. They're very flat, large cells. Actually, in this case, it's onion skin and buccal cells, which are cheek cells. So you can prepare a sample like this just by swabbing the inside of your cheek and smearing it onto a glass cover slide. You'll usually find some cells like this. So, I mean, you can see already that we've got a bit of structural information just from these images. You can see inside each cell, there's perhaps something that you could convince yourself looks like a nucleus, a nucleolus. So the light microscope is great for this kind of structure. But what we're interested in is how high resolution can we go? You know, we want to see things at the atomic level. And actually, when we talk about what's the smallest thing we can see, what we're really talking about is resolution. So a kind of loose definition of this is how much do you need to separate two items so that you can tell that they're two distinct things? So it sounds like a trivial question, but actually in microscopes, that's quite a complicated thing to figure out. But, you know, 100 or so years ago, had Ernst Abbe, who came up with his theory of diffraction limited resolution, which basically lets us put into a mathematical formula for a light microscope what's the smallest thing that we can see. And it turns out that that's related to this parameter that's on the top, lambda. It's a Greek letter that we usually use to represent wavelength. And then on the bottom of the equation, we've got 2 times the refractive index, which for glass or for water, something like this, and sine alpha, and alpha is a constant that's kind of related to the size of the lens and how good it is at capturing all that light that's coming into it. And these things set the resolution limit in the microscope. Now, in an optical microscope, because of the limitations that we have on refractive index and how well we can make glass lenses, actually, we can just dumb this equation down into saying that, well, about the smallest thing that we can see, the resolution D, is about the wavelength of light. Now, if you think back to your physics classes and what the wavelength of light usually is about, it means that D is about 0.2 to 0.5 micrometers. So in this case, 1 micrometer or 1 micron is 10 to the minus 6 meters. So it's a millionth of a meter, really small. And that's why we can get such good detail even in our basic optical images, because we're already looking at with a tool that's capable of resolving tiny objects. And in biology, this is a pretty good starting point. Our microscope resolution is plenty good enough to be able to distinguish mammalian cells. So these are typically on the order of about 10 microns is normal, one's really small, 100 is getting really big. They're somewhere in that range. Viruses are much smaller, they're about 20 nanometers to maybe 400 nanometers. So really, you know, a few 100 times smaller than a cell. And individual proteins are macromolecules. So all of the sort of atomic structure that makes up things like a virus, makes up things like DNA. When we're talking about wanting to look at single particles like that, what we're really talking about is things that are on the order of just a few nanometers. So that's 10 to the minus 9 meters. So we've gone down even 2 orders of magnitude there in scale from the size of a single cell. So at that point, when we're starting to get into this regime of wanting to look at viruses, proteins, even things like organelles within the cell itself, you can see I've put this image up here that shows all of the things inside a plant cell that we can't see necessarily with a light microscope, like mitochondria, vacuole, things like this. We really need to find a way to look smaller. And we've already said, well, the light microscope, the resolution limit is due to the wavelength of the light that we use. So we can't really do a whole lot better. So what are our options? We think about Ernst Abbe's equation again. Well, the most powerful tool that we've got in that arsenal actually is to change the illumination wavelength. So we change the top term on that fraction and really drive down our resolution limit so that we can see smaller and smaller. This is kind of illustrated by if you take an electron. So here I've drawn a little schematic of an electron or 2 electrons orbiting around an atomic nucleus. If you rip those electrons off, focus them down into a beam, actually their wavelength is about 6 times 10 to the minus 16 meters. So if we're then in that diffraction limited resolution limit, where D is approximately lambda, what we're saying is we can look at something really, really tiny, much smaller even than an atom, and definitely smaller than a cell, virus, protein, anything like that. Now, in practice, it's a little bit more complicated than that, because it turns out we're not so great at making really good electron microscope lenses, certainly not as good as we can focus down light that's passing through a glass lens, but we can still do really well because this wavelength is so short. So how this looks in the lab is actually you scale up the microscope massively as you scale down the resolution limit. So you can see I tried to indicate little benchtop microscope sitting on the floor, and our high resolution electron microscope takes up a whole room. Actually, what you don't see in that image is the fact that that room is about 4 times the size of the microscope, because we've got all kinds of other bits of kit in there. And we have a second room that has to contain all of the cooling, all of the power generation, all of the computers. So it's a really big bit of kit. But actually the operating principles are fairly similar, because before we drew this illustration where we have an illumination source at the bottom with photons passing through some lenses, passing through our sample, collect what's transmitted. Actually, it's really similar in the electron microscope. Apart from you swap your illumination source from photons to electrons, your lenses are no longer made of glass, they're electromagnetic coils. You can't use a glass cover slide. Instead, we use a TEM grid, which is like a very, very thin piece of metal, sometimes with some carbon film, and we put the sample on there. And then we collect all the electrons that come through. So like it says on the slide here, the key differences are really in the lenses and then in the sample preparation and how we need to get things on there. So in an optical microscope, you can just collect a sample quite easily. Like I say, swab the inside of your cheek, peel off some skin from an onion, put it onto a glass cover slip, and you're kind of good to go. Maybe you might add some stain. In the TEM or in the electron microscope, it's much more intensive than that, because you can't pass electrons through very much material because they just tend to get absorbed. So what we have to do is try and find a way of thinning our sample down so that it's less than a couple of 100 nanometers thick. we also have some difficulties depending on the specific type of sample in how we can get that into the microscope, which I'll talk a bit in a second. And the last point is about size and cost. You need a whole room to fit in, and they're really expensive. So it tends to be dedicated facilities like the one that we've got at Oxford, where you can do this really high resolution work. So this is an example then of the kind of images that you can get out of an electron microscope. So in this case, these are images taken from a platinum-based chemotherapy drug called cisplatin, dispersed onto one of those TEM grids that I was talking about. And when you zoom in, and you can see the scale bar on the images on the right-hand side is 2 nanometers, you can see actually these regular arrangements of dots, right? And actually what you have there is each dot being a column of atoms that the microscope can resolve from a separate column of atoms. And those 2 columns of atoms are spaced about 0.4 nanometers apart. So what you've got there is an atomic resolution image of platinum inside a drug that's administered to cancer patients. This is super powerful because from images like this, you can actually, with a lot of maths, go and construct 3D models of how that nanoparticle looks in real life. Because what you're seeing here is actually just a projection of that nanoparticle onto an image. But because the way that the image is formed is quite complex, you can back propagate that and come up with 3D structure. And of course, this is something that would be really nice to be able to do in biology, right? Like what we want to do a lot of the time is try and understand how things fit together and how structure influences function. So that's what I'm going to talk about for the rest of the presentation. So in biology, the main challenges that we've got with getting a sample in is actually that organic matter has lots of quite weak bonds. And these bonds are really easily broken when you hit them with high energy electrons like we have to do in the microscope. I already mentioned that you can't use a glass cover slip in a microscope, and that's primarily because it's way too thick. The electrons are going to get absorbed in there, so you need to make sure that your sample's super thin, which you can probably imagine already is going to be a problem if you want to look at a tissue sample that's been biopsied out. You need to find a way to get that to be less than a couple of 100 nanometers. Single proteins, they're much smaller, you know, maybe a few nanometers already, but you need to find a way of purifying them so that you can get them onto a grid and only look at that protein. So it's all quite complicated stuff. The biggest problem of them all, water. Just like you can't get electrons through loads of material, you also can't get them through loads of air. So we have to keep the microscope under really high vacuum, and that is the biggest enemy of water. Cells really do not like to be in that environment. No biological matter does. So we have to come up with clever ways to try and fix that water in place so that the sample doesn't just sort of crumble under the electron beam. Actually, when you figure out how to do that, it turns out you get a Nobel Prize, which is quite nice. So in 2017, the prize was awarded to Jacques Dubochet, Joachim Frank, and Richard Henderson, who all played a massive part in developing what's now a huge field in electron microscopy and a subfield called cryo-electron microscopy. And their big revolution was finding that if you freeze biological tissue slowly, you form crystalline ice where all of the atoms are in a regular arrangement, and this disrupts the biological material massively. So it causes all of the cell membranes to rupture, the contents of the cell to leak out, and you can no longer relate structure to function because you've destroyed your sample. So their big thing was finding actually the solution of that is to freeze really fast, because if you manage to do that, you don't give the atoms in the water enough time to arrange into that regular pattern. You just kind of capture them in place. So they developed this technique like plunge freezing, where, for example, you spray material onto a grid as you plunge it into liquid ethane, and you freeze it in literally fractions of a second. And this gets around that problem of disrupting all of the structure that's really important to our ultimate goal in research, to try and understand how this stuff works. So when you manage to do that, this is a really nice example of single particle CryoEM, where you have put a protein called GROL onto a TEM grid, plunged it into liquid ethane, frozen it in place. And what you can see here, I hope it's okay to see on the screen, is that you've got individual proteins dotted around on your sample, and they're embedded in this vitreous water, this amorphous water from freezing really fast. So they're as close to their native environment as possible. And if you zoom in a little bit, you might be able to convince yourself. You can see some things that look a little bit like a stripy rectangle, and a little other configuration that looks like a kind of bumpy circle. And we're not at atomic resolution yet. So the diameter of this CryoEM protein is about 140 nanometers. And what we'd really like to do is try and up the magnification or up the resolution so that we can see individual atoms in this protein. But unfortunately, because of the problem I talked about before, which is these bonds being quite weak, we can't just hit them with more and more electrons and try and zoom in, because we're just going to end up blasting this little protein apart. And the whole thing of the Nobel Prize was, wouldn't it be cool if we could find a way to use this kind of low magnification, lower resolution image and figure out atomic structure? So it turns out with a lot of images, a lot of configurations, you can identify in thousands of images like the one on the left, different arrangements of the same protein. And then again, with a lot of maths, kind of work backwards. And out of this, pull out this kind of structure. This paper, I think, was actually published just a couple of weeks ago. It's a very new paper. But what you have is, from this image and using kind of the power and the resolution of the electron microscope, the ability to resolve really tiny features inside proteins. And again, from those types of images, with a lot of modeling and a lot of computational work, you can figure out exactly what types of atomic arrangements are where. You can see some coiled structures, for example, on the outside. And from here, try and understand how this protein works. Now, CryoEM is actually a really cool little protein because it's involved, it's found in bacteria, and it's involved in all kinds of folding of other proteins. So its role is really to help other macromolecules fold into the right shape so that then they've got the right structure to go on and do their jobs. And it works in combination with another protein called CryoS, which kind of sits on the top of this little tube like a hat. And then all the protein folding occurs inside in the cavity, so in the hole that you can see on the bottom right image. So it's a really nice way that we can kind of look at these proteins at super high resolution and then figure out how they work in combination with other proteins to sort of do their jobs. And I mean, it's pretty topical at the minute to talk about COVID, so why not? You can see here a graph actually of structures from cryo-electron microscopy deposited in this online data bank. So it's the EMDB in time. And for COVID, if you go across from 2019, that's when people maybe start talking about it. 2020, you've got a lot, and now we're at over 500 structures, which is a huge amount of work in not very much time. So, I mean, I just wondered if we've got time to ask a question. Why do you think people are so interested in knowing what the shape of the SARS-CoV-2 is? You know, we talked a bit in CryoEM about how the structure and how being able to see it at atomic resolution lets you understand function. So what about for COVID? Yeah, so someone says it looks a bit like the protein we saw before. Kind of. So they don't do exactly the same job. But it's all about this idea of, if we can see how the virus looks, how it looks to our immune system, maybe we can come up with ways of making our immune system recognize it before we're infected. And anyone who's still listening kind of knows that I'm going to be talking a little bit about vaccines. This is exactly it, right? So we can take COVID virus particles, disperse them on a grid, freeze them so that we capture all that water, we don't disturb their native structure. And actually in CryoEM, then you can start seeing these little proteins that are stuck on the surface of the virus. And using the exact same techniques that are used for CryoEM to understand that structure and potentially function, can do exactly the same with the proteins that are stuck on the outside of the COVID virus. And when we resolve that to really high resolution, again, we can start seeing, okay, this is how it presents. What do we need to do to make sure our immune system knows what this protein looks like? Because the presence of this protein means COVID infection. So when we develop vaccines and when we talk about things like mutations and that leading to new variants, generally what we're talking about is mutations in these proteins that are on the exterior of the cell. Because the more different the protein looks, the less well your immune system can recognize it. So like I say, it's kind of more relevant now than ever to be able to see particular biological structures at really high resolution so that we can detect any changes like this. So like I say, in summary, we've sort of discussed that light microscopes can image really small things, definitely smaller than a cell, but electron microscopes can do it even better or at even higher resolution. But as a compromise, there are lots of challenges with biological materials, particularly because of the water. So we've got to make use of some clever imaging techniques, lots of maths to try and get us back to something that's high resolution or especially atomic resolution. So that we can then take this information and apply it to kind of our understanding of how fundamental biological processes work. But it's a really cool field. I'm happy to take any questions that you guys have got now, because yeah, I hope you've enjoyed it. Thank you.
Brilliant. Thank you so much, Alex. That was such a fascinating talk. I really enjoyed listening all about how we've gone from light microscopes to electron microscopes in order to see much, much smaller things. And it was also really cool to see some actual images there from the electron microscope, particularly of the SARS-CoV-2 virus. That's really interesting to see.
Yeah,
Okay,
it's
so
really cool. There's been so much work done on that in the last 18 months. It's been absolutely incredible.
Yeah, it's been a huge effort from scientists, hasn't it, to get all of that work done in such a short amount of time. It's been incredible.
Absolutely colossal, yeah. I think there's a question actually. So what's the hardest part of cells to see under a microscope? That's a really good question, actually. So one of the things that we struggle with in the microscope is generating enough contrast to be able to distinguish stuff. And contrast is just like in a photograph. It's how light or dark something is compared to the stuff that it's kind of embedded in. So anything that's got a similar density is normally a bit tricky to spot amongst a field of other stuff that's a very similar density. So what we try and do a lot of the time is add things like stains. So in the case of the light microscope, that's stuff like methyl blue. It's stuff that stains specific parts of the sample that you're interested in. So you can get specific stains for membranes, for DNA, for proteins. And we also have correspondingly in the electron microscope stains that we can add. They're usually made of heavy metals, but they selectively stain as well. So stuff like uranyl acetate, osmium tetroxide, they're really nasty metallic stains. But because the cells are already dead at that point, they're useful to have in your arsenal to be able to see a little bit more detail.
Brilliant. Yeah, that's really interesting.
So there's a question of how can you see biological material under the TEM grid? You mentioned it's made from metal. Is it so thin it's transparent? Yeah, it's a really good point. So the grid is metal. And then what you do is between the bars of the grid, so it's kind of mesh, you suspend something like very, very nanometer thin carbon and then put your sample on that. So that's just about thin enough that it doesn't disturb your image too much without adding lots of thickness. But some people use really weird supports as well. If you, especially in material science, you can put things on like silicon nitride, graphene, stuff like that. It just depends on your application, but for biological materials, it's generally very thin carbon or very thin polymer. And then someone else asked, does this count for solids and gases? I'm not sure quite what that means. If it means, can you do electron microscopy on solids and gases? Definitely on solids. And also some people are doing some really interesting work in trying to do microscopy on things like liquids. So, and even liquids that you then can sort of heat up, cool down, pass an electric current through in the microscope and see the associated changes. So actually Emma, who's doing the webinar next week, might talk a little bit about that, because she's going to be talking about battery materials, I think, and something that they do quite often is actually look at sort of charging and discharging battery materials in the microscope so that you can see the atomic structure change with the charging and discharging. And it's a really cool application.
It sounds very tricky as well.
Oh yes. All of this work is really, really difficult. It's the reason that there's been so many Nobel Prize awarded for electron microscopy, because it's just, yeah, it's really difficult, but it's a lot of fun.
Groundbreaking new leaps.
Yes, definitely. Well, like I say, that the model of the COVID spike protein I showed on the last slide, I think it was published on the 24th of November. So it's kind of like a really, really up-to-date science. Yeah,
absolutely. So brilliant. I think we probably have time for one more quick answer. I know we've run over slightly, but we did have the technical hitches at the start, so I hope you don't mind running over just by 5 minutes. But
there's a great question about actually how to use the low resolution images to know the molecular structure by a lot of maths. Yeah, it's a good question. So when you use those low resolution, low magnification images, what you rely on is the fact that when you've dispersed your single proteins onto the TEM grid, they're kind of in all different random orientations. So you actually have a full 3D view of the structure. It's just, rather than you looking in 3D, it's kind of lots of different views from lots of different directions spread all across the image. So it's an absolutely colossal task to categorize those images. So obviously, because we're talking about tens of thousands of images and often millions of individual proteins, we don't do that by hand. We write really complex algorithms to try and categorize it, but it's super difficult because what you have to do is figure out, okay, this protein that I'm seeing, what orientation is it in? Is it definitely the protein that I'm interested in? And then once I am sure that it yes to both of those questions, how do I then incorporate that into my 3D model so that I know exactly where it fits in the real shape of the molecule? It's really difficult, but fortunately we've got lots of very good computational physicists, computational biologists who can work on this.
And that just really highlights all the scientists collaborating together as well. We've got the biologists, the physicists, material scientists, computer scientists, so they work together. I mean,
I always think the lines get very, very blurry at this point, because what you need to do this kind of work is proper collaboration between physicists, chemists, biologists, computer scientists, engineers. The list just goes on and on. So it's really really exciting field. Yeah, it's really interesting.
Yeah, absolutely. And on that note, I think we'll wrap things up. So thank you so much, Alex, for giving us such a fantastic talk and answering all your brilliant questions. And thank you all for joining us, and have a lovely evening, everybody.